Hello everyone, my name is Kelsey Bond and today I'll be presenting on Huntington's disease and the genetic basis of neurodegeneration. When I was choosing a topic for this presentation, I really wanted to learn about a disease that I didn't know much about but had heard of before. As I started researching Huntington's disease, I found it incredibly interesting that a change in just one gene can have such a profound impact on a person's brain, body, and even future generations of a family. Throughout this presentation, I'll explain what Huntington's disease is, how a mutation in the HTT gene causes the disease, how it is inherited, and some of the exciting research that's being done to better understand and treat it. To begin, Huntington's disease is a progressive neurodegenerative disorder. In simple terms, that means the nerve cells in the brain gradually become damaged and die over time. This disease affects movement, thinking, and behavior, and although the genetic mutation is present from birth, symptoms usually don't begin until adulthood, often between the ages of 30 and 50. One thing I found especially interesting while researching this disease is that a person can carry the mutation for decades before they ever notice symptoms. By the time symptoms appear, the disease process has actually been occurring for many years. As shown in the timeline on the right, Huntington's disease progresses through several stages. It begins with an at-risk stage where the mutation is already present but no symptoms are visible. As the disease advances, people may first notice subtle changes in mood or coordination, followed by increasing movement difficulties, cognitive decline, and eventually severe disability. Although there is currently no cure, researchers continue to study Huntington's disease in hopes of developing treatments that can slow or even stop its progression. As I continued on with my research on Huntington's disease, I realized that nearly every aspect of the disease can be traced back to one genetic mutation. That's what made this topic just so fascinating to me. The central focus of my presentation is that Huntington's disease is caused by an expansion of CAG repeats within the HTT gene. This mutation produces an abnormal Huntingtin protein that gradually damages neurons in the brain. The disease follows an autosomal dominant inheritance pattern, meaning it can be passed from one generation to the next with a 50% chance if one parent carries the mutation. Throughout the rest of this presentation, I'll explain how this mutation affects the brain at the molecular level, how the disease is inherited, and why current genetic research offers hope for future treatments. This slide illustrates the molecular genetics behind Huntington's disease by focusing on the HTT gene. The HTT gene is located on chromosome 4, specifically at position 4p16.3, and normally produces the Huntingtin protein. Under normal conditions, the Huntingtin protein plays an important role in neuron survival, protein transport, gene regulation, and other essential cellular functions within the brain. The critical mutation occurs within exon 1 of the HTT gene, where a DNA sequence called a CAG repeat is located. Normally, the HTT gene is first transcribed from DNA into messenger RNA. The messenger RNA is then translated by the cell to produce the Huntingtin protein. Each CAG repeat codes for the amino acid glutamine. When too many CAG repeats are present, the Huntingtin protein develops an abnormally long polyglutamine tract, causing the protein to fold incorrectly. The misfolded protein begins forming Toxic clumps inside neurons. These protein aggregates interfere with normal cellular functions, disrupt gene expression, impair energy production, and eventually cause nerve cells to die. The neurons most affected are located in the striatum, which explains why Huntington's disease causes progressive movement disorders, cognitive decline, and behavioral changes. This single genetic mutation ultimately leads to the symptoms seen in patients with Huntington's disease. As you can see, this figure compares a normal HTT gene on the left with an HTT gene affected by Huntington's disease on the right. In a healthy individual, the HTT gene contains 26 or fewer CAG repeats within exon 1. These repeats produce a Huntington protein with a normal length polyglutamine tract, allowing the protein to function properly within the neurons. In individuals with Huntington's disease, the number of CAG repeats expands to 40 or more repeats. This expansion creates an abnormally long polyglutamine tract within the Huntington protein. As the Huntington protein becomes longer, it begins to misfold and lose its normal shape. These misfolded proteins accumulate inside neurons and interfere with essential cellular functions. Over time, these toxic protein aggregates damage and eventually kill nerve cells, particularly those within the striatum of the brain. This progressive neuronal loss is what leads to the movement disorders, cognitive decline, and behavioral symptoms characteristic of Huntington's disease. Now that we've talked about how the mutation changes the Huntington protein, let's look at what actually happens inside of the neuron. As you can see here on the far left, this is the mutant Huntington protein. Because of the CAG repeat expansion we discussed on the previous slide, this protein is longer than normal and doesn't fold correctly. As you can see here, these dark spots are protein aggregates. They're made up of many misfolded mutant Huntington proteins that have clumped together inside of the neuron. These proteins aggregate are toxic because they interfere with the cell's normal functions. Moving over to the next image, you can see a damaged neuron. As these protein aggregates continue to build up, they disrupt gene expression, protein transport, and energy production, causing the neuron to become stressed and damaged. Finally, as you can see in this last image, the neuron eventually dies. Huntington's disease primarily affects neurons in the striatum, which is why patients gradually develop problems with movement, thinking, and behavior as the disease progresses. So this entire process begins with one genetic mutation, which leads to an abnormal protein aggregation, neuronal damage, and ultimately the symptoms of Huntington's disease. Now let's take a look at how Huntington's disease is inherited from one generation to the next. As you can see here, the father is affected by Huntington's disease because he carries one mutated copy of the HTT gene. Since Huntington's disease is an autosomal dominant disorder, only one mutated copy is needed for a person to develop the disease. The mother is unaffected, meaning she has two normal copies of the HTT gene and does not have Huntington's disease. When these parents have children, each pregnancy is independent and each child has the same chance of inheriting the mutation. As you can see here, each child has a 50% chance of inheriting the mutated HTT gene and a 50% chance of inheriting two normal copies of the gene. If a child inherits the mutated HTT gene, they will likely develop Huntington's disease later in life. If a child does not inherit the mutation, they will not develop Huntington's disease and cannot pass the mutation on to future generations. These autosomal dominant patterns explain why Huntington's disease often appears in multiple generations of the same family. In this illustration, these children represent the possible outcomes. This does not mean that every family will have exactly 2 affected and 2 unaffected children. Each child has an independent 50% chance of inheriting the mutation. Now let's take a look at where Huntington's disease, or HD, is common around the world. As you can see here, HD is most common in Europe and North America, where it affects about 3 to 7 people per 100,000. In contrast, HD is less common in Asian and African populations. Researchers believe that this is because expanded HTT gene mutations occur less frequently in these populations. Genetic testing can identify people who carry the HTT mutation, helping families make informed decisions and allowing researchers to study the disease more effectively. Overall, studying HD in different populations helps scientists better understand who is affected and why. Although there are still no cures for HD, researchers are making important progress towards new treatments. As you can see here, gene silencing therapies are designed to reduce the amount of mutant Huntington protein. The second panel shows antisense oligonucleotides, or ASOs. These treatments target the HTT messenger RNA to reduce production of the harmful protein. The third panel shows CRISPR gene editing, which may one day allow scientists to directly target the HTT gene. Finally, a 2025 study by Hansucker and colleagues found that CAG repeats can continue expanding in brain cells over time, which may contribute to disease progression. Together, these advances give researchers hope for developing treatments that may slow or even prevent the progression of HD. In conclusion, HD is caused by a mutation in the HTT gene. As we've seen, this mutation causes CAG repeat expansion, leading to an abnormal Huntington protein. Over time, this abnormal protein damages neurons, causing the movement, cognitive, and behavioral symptoms associated with HD. Although there is currently no cure, ongoing research continues to improve our understanding of the disease and offers hope for future treatments. As research continues to advance, scientists move closer than ever to developing therapies that may slow or even prevent the progression of HD. Thank you.